Coming to next metabolism, amino acid metabolism. So in this first we are going to see about what is the fate of amino group in an amino acid. So when you see structure of amino acid, so amino acid has two functional group, amino group and carboxyl group. So when amino acid undergoes catabolism, the first step in the catabolism of amino acids is removal of amino group. So here we will discuss about what happens to that amino group after its removal, and how it is removed and how it is transported to the liver. Right? So that is metabolism of ammonia. Right? So ammonia is formed in the catabolism of ammonia amino acids by two mechanisms, that is transamination and oxidative deaminations. So in this way, the amino group in amino acid is transferred to the liver by transamination and oxidative deamination. Right. So first we will see, so these two are coupled process, together we call it as transdeamination. So as I told you, the amino acid in its catabolism loses the amino group, so we have to remove the amino group from the amino acid. So in that removal, first the amino group is transferred to a keto acid, so that keto acid is converted into amino acid, and that amino acid is transported to the liver. So this is what basically happens in the removal of amino group from the amino acids. So first mechanism that occurs is transamination. So transamination can be defined as transfer of amino group from amino acid to a keto acid. So that results in the synthesis of a new amino acid and keto acid. So this is how the amino group in one amino acid is transferred to a keto acid, and the keto acid which has received the amino group is converted into a amino acid, and the amino acid which transferred the amino group is converted into keto acid. So that keto acid now enters into the catabolic pathway of amino acid, and after receiving the amino group, that keto acid which is converted into a new amino acid is transferred to the liver, is transported to the liver. Right. And most of the amino groups in the tissues, they are carried by glutamic acid. So alpha ketoglutarate acid accepts the amino group and it is converted into glutamic acid. So any amino acid, it loses the amino group, it transfers the amino group to alpha ketoglutarate acid, so that results in the synthesis of glutamate. So glutamate is formed from the alpha ketoglutarate acid when it is receiving the amino group. That glutamate now from the peripheral tissues is transported to the liver. So most of the, when you see the concentration of amino acids in the blood, so when you compare the concentration of glutamic acid to that of other amino acids, so 10 times more amount of glutamic acid is seen than the other amino acid, because it is involved in the transport of amino group from the peripheral tissues to the liver. So major form in which amino group is transported is glutamic acid, and when you see in case of brain, and that glutamic acid, that amino group in the brain is transported in the form of glutamine. So glutamic acid reacts with amino group and it forms glutamine, and that glutamine from the brain reaches to the liver. So the form in which amino group is transported from the brain to the liver is glutamine, and the form in which amino group is transported from other tissues to the liver is glutamate. So the concentration of glutamine and glutamate is more in the blood when compared to other amino acids. So this is how transamination transfers the amino group to the keto acid, and that keto acid is converted into glutamic acid. And what happens to that glutamic acid? That glutamic acid, and one more thing, those transamination reactions are catalyzed by a group of enzymes called as transaminases or amino transferases. And that glutamate, now after reaching the liver in hepatocytes, it undergoes another process called as oxidative deamination. So transamination simply involves the transfer of amino group. So that happens in the peripheral tissues. Now, once glutamic acid reaches the liver in the hepatocytes, it undergoes oxidative deamination. So deamination is removal of amino group, and that is coupled with oxidation. So that's why we call it as oxidative deamination. right? So here we require the coenzymes of niacin, that is NAD plus and NADP. NADP plus are required in this oxidative deamination, and there is one enzyme called as glutamate dehydrogenase that can use NAD plus or NADP plus as the coenzymes. Any one of them can be used by this enzyme, and that enzyme catalyzes the oxidative deamination of glutamate into alpha ketoglutarate acid with the removal of amino group. So amino group, which is transferred by the amino acids in the peripheral tissues, is now released in the hepatocyte, is released in the liver by the action of enzyme called as glutamate dehydrogenase. Now, this is how the amino group is transferred from the peripheral tissues to the liver. It undergoes oxidative deamination and the amino group is released in the hepatocytes. And what happens to that amino group? Right. So that we'll discuss in next topic. So now we can see that these two processes, transamination and deamination, both are coupled process. So that's why we call it as transdeamination. Though we'll discuss it in a separate. So it is actually transamination coupled with deamination. Transdeamination occurs. Amino group is transferred in the peripheral tissues, is released in the liver in the form of amino group by the action of enzyme called as glutamate dehydrogenase. Right? So that process we call it as oxidative deamination. So what happens to the ammonia in the liver? So ammonia is toxic compound, it is a neurotoxic compound. So we have to detoxify that ammonia, right? So that detoxification of ammonia occurs in the liver. So when you see in case of brain, as I told you, glutamic acid takes up the amino group and is converted to glutamine, and that releases the amino groups in the liver by the action of enzyme glutaminase. So glutaminase acts on glutamine, which is transported to the liver from the brain, and that glutamine having the amino group is released by the action of enzyme called as glutaminase. Right? And coming to detoxification of ammonia. So that ammonia which is released in the hepatocytes now is as I told you, it is a toxic compound. We have to convert that toxic compound into non-toxic compound. For this conversion of the neurotoxic ammonia into a less toxic compound and more water-soluble compound, we require a help of the cycle called as urea cycle. So this conversion of ammonia into a less toxic compound, urea, is brought by a particular cycle called as urea cycle. So what happens in urea cycle? In urea cycle, whatever the ammonia which is released in the hepatocytes is converted into the whatever the ammonia which is released in the hepatocytes is converted into urea. So what is urea? Urea is the end product of protein metabolism. So that's how we can define urea. Urea is the end product of protein metabolism. And where this urea cycle occurs? So urea cycle occurs in the liver. And when you see, so the enzymes concerned with the urea cycle, they are located both in the mitochondria and cytosol. Right? So major organ where urea is synthesized is the liver. And coming to reactions of urea cycle, so as I told you, the reactions they occur in both cytosol and mitochondria. So the ammonia which is released in the liver mitochondria is first undergoes, it condenses with, it reacts with carbon dioxide, and this reaction is catalyzed by the enzyme called as carbamyl phosphate synthetase 1. So there are two carbamyl phosphate synthetases. So carbamyl phosphate synthetase 1 is involved in the urea cycle, urea synthesis. So this CPS1, in short we call it as CPS1, catalyzes the condensation of carbon dioxide and ammonia, and that results in the formation of carbamyl phosphate synthase. Right? So carbamyl phosphate synthase, which is formed, so carbamyl phosphate is formed by the action of enzyme carbamyl phosphate synthetase 1. So this reaction requires the energy supplied by ATP. So here we require 2 ATPs. 2 ATPs are hydrolyzed to 2 ADPs, and that results in the formation of carbamyl phosphate by the action of enzyme CPS1. So this carbamyl phosphate formation occurs in the mitochondria, and the next reaction is, so this carbamyl phosphate reacts with ornithine and forms citrulline. So this is catalyzed by an enzyme called as ornithine transcarbamylase. So ornithine and carbamyl phosphate react to form citrulline, and this is catalyzed by ornithine transcarbamylase, and this reaction is also occurs in the mitochondria. So first two reactions in the urea synthesis, they occur in mitochondria, and citrulline which is formed in the mitochondria is now transported out of mitochondria into the cytosol by specific transporters. So citrulline transporters, they transport citrulline out of mitochondria into the cytosol. So that citrulline in the cytosol reacts with aspartate and gets converted into arginosuccinate, right? So citrulline and aspartate react, and thus this reaction is catalyzed by the enzyme called as arginosuccinate synthase. And this enzyme also requires, this reaction also requires energy, where 1 ATP is hydrolyzed into AMP, right? So ATP is converted into AMP. So this reaction is driven by the utilization of ATP. So one molecule of ATP is converted into AMP. So that citrulline is converted into arginosuccinate. Arginosuccinate in the next reaction catalyzed by the enzyme called as argininosuccinase. So that arginosuccinase removes fumarate from the arginosuccinate, so that arginosuccinate is converted into arginine. And what is the fate of arginine? So in the next reaction catalyzed by the arginase, arginase acts on arginine and it converts arginine into urea, and it regenerates the ornithine. So that is the action of arginase on arginine, right? So this is how again ornithine is regenerated, and ornithine again reacts with one more molecule of carbamyl phosphate. So this cycle occurs continuously. So this is how whatever the ammonia which is released in the hepatocytes is converted into a less toxic compound, urea. So there are 5 reactions in the urea cycle, so that convert ammonia into urea. So urea is less toxic when compared to ammonia, and it is more water soluble, and it can be easily excreted through the urine. So that is the significance of urea cycle, and that is how ammonia is detoxified in our body by a cycle called as urea cycle. And when you see, there are some important disorders concerned with the urea cycle, and as I told you, there are total 5 reactions in the urea cycle. So whenever there is block in the reaction of any reaction of the urea cycle that leads to a particular disorder, we call collectively we call all those disorders as urea cycle disorders. So when you see, whenever there is deficiency of first enzyme, that is which catalyzes the condensation of ammonia and carbon dioxide, that is carbamyl phosphate synthetase 1, it leads to, whenever there is block in this reaction because of deficiency of this enzyme, it leads to a condition called as hyperammonemia type 1, right? So whenever there is deficiency of second enzyme, that is ornithine transcarbamylase, it leads to a condition called as hyperammonemia type 2, another urea cycle disorder. And whenever there is defect in the transporters which transport citrulline from the mitochondria into cytosol, so then it leads to increased citrulline concentration in the blood, and that is called as citrullinemia, right? So that's you can see. And whenever there is deficiency of enzyme arginosuccinate synthase, so arginosuccinate synthase deficiency is there, then also it leads to increased concentration of citrulline, right? And when you see other enzyme deficiency, arginosuccinate lyase, arginosuccinase. So whenever there is deficiency of arginosuccinase, then the concentration of arginosuccinic acid increases in the blood, and that condition we call it as argininosuccinic aciduria, right? So that's about the enzyme defect arginosuccinase, and it leads to a condition called as arginosuccinic aciduria. So it is acid and it is excreted in excess in urine, we call it arginosuccinic aciduria. And when you see, whenever there is deficiency in the enzyme arginase, so that arginase deficiency leads to increased concentration of arginine in the blood, we call it as argininemia. So this is how deficiency of each enzyme in the urea cycle leads to a particular urea cycle disorders. So collectively we call all those disorders of urea cycle as urea cycle disorders. Yeah. Right. So that's about urea cycle. So we have discussed how the ammonia is transported from the peripheral tissues to the liver, how it is released in the liver, and how it is detoxified by the urea cycle, and what are the urea cycle disorders. Now coming to inborn errors of amino acid metabolism, here you can see the most important one is phenylketonuria. So the name itself indicates that there are excess phenylketones excreted in the urine. Phenylketonuria. So all of them, they have a keto group, phenylketones, and they are excreted in phenylketonuria. So we call it as phenylketonuria. In short, we call it as PKU. Coming to what causes the phenylketonuria. The enzyme deficiency in case of phenylketonuria is phenylalanine hydroxylase. So that enzyme actually converts phenylalanine to tyrosine. So whenever there is block in that reaction due to deficiency of this particular enzyme, phenylalanine hydroxylase, it leads to increased concentration of phenylalanine. So that phenylalanine in excess is converted into another compounds called as phenylketones. So it will be converted into phenylactate, it will be converted into phenyl pyruvate, and it will be converted into phenyl acetate. So like that, it is converted into phenylketones. So phenylalanine cannot be converted into tyrosine. So phenylalanine is undergoes transamination, is converted into phenyl pyruvate, and that will be converted into phenyl acetate and phenylactate. So all of them are ketones. So there is excess phenylketones in the body because of deficiency of this enzyme. That's why we call it as phenylketonuria. Right. And coming to it, incidence. So this is seen. So 1 in 10,000 births. For every 10,000 births, you can see one case of phenylketonuria. So that is incidence of PKU. And coming to biochemical features, what happens in phenylketonuria? As I told you, there is increased concentration of phenylketones. So like that, phenyl pyruvate, phenyl acetate, phenylactate, all of them are increased in the blood. Coming to clinical manifestations, what happens in case of phenylketonuria? So there is mental retardation, that is the most, and so apart from that, there is tremors, convulsions, and hypopigmentation. Right? These are the important clinical features of clinical manifestations of phenylketonuria. And coming to diagnosis, how can you diagnose these cases? So this can be done by chromatography, paper chromatography, Guthrie test is there, and a simple test, ferric chloride test, is also used in the diagnosis of phenylketonuria cases. And apart from that, what is treatment for this phenylketonuria? This phenylketonuria can be treated by decreasing the amount of phenylalanine in the diet. So you have to decrease the intake of phenylalanine, and for the and in case of phenylketonuria, the prescribed amount of phenylalanine should be taken per day is, it should be 10 to 20 milligrams per kg body weight. So that should be taken, and when you take that much amount of phenylalanine, you can prevent the phenylketonuria. Right? And coming to another inborn error of amino acid metabolism, alkaptonuria. So this is because of deficiency of an enzyme called as homosensate oxidase. And when you see, and this is inherited as autosomal recessive condition, it is inherited as autosomal recessive, and incidence is less than the phenylketonuria. So it is 2 to 5 per million births is seen in case of alkaptonuria. Right? So enzyme deficiency is homosensate oxidase. So that enzyme deficiency leads to alkaptonuria. And what is the biochemical manifestations or features of alkaptonuria? So whenever there is deficiency of homosensate oxidase, the concentration of homogensic acid increases in the blood, and that gets oxidized, and that homogensic acid gets deposited in the tissues, and it leads to blackening of the tissues, and that is seen mainly in case of alkaptonuria. So blood and urine, so it is the excess homogensic acid is seen in blood, and that is excreted in the urine. And when you see, when you collect that urine and expose it to air, then what happens? the blackening of urine occurs from top to bottom because of oxidation of that homozynic acid. Right? So that is the important feature in case of alkaptonuria. So blackening of urine occurs when you expose it to air because of oxidation of homozynic acid. So that is how it leads to dark urine on standing. And when you see in the metabolism of phenylalanine tyrosine, deficiency of each enzyme leads to a particular type of inborn errors of metabolism. And when you see in the conversion of phenylalanine to tyrosine, it leads to phenylketonuria. And in the next enzyme, whenever there is deficiency in the enzyme, which converts tyrosine into parahydroxyphenyl pyruvate and tyrosine transaminase, that enzyme deficiency leads to tyrosinemia type 2. So like that, when you see alkaptonuria, that is because of deficiency of enzyme homozynic oxidase. So like that, each enzyme in the metabolism of phenylalanine and tyrosine leads to a particular type of inborn error of amino acid metabolism. Okay. So these are the most important feature which can be explained in case of alkaptonuria is ochronosis. So what is ochronosis? So whenever that homozynic acid gets deposited in the tissues, it leads to blackening of those tissues. So that blackening of tissues and is called as ochronosis. Generally, homozynic acid gets deposited in the joints and that leads to arthritis. Apart from that, the most important feature is, so when it gets deposited in the intervertebral disc, so you can see degeneration of that intervertebral disc because of deposition of homozynic acid, and it gets deposited in the cartilages and all the soft tissues it gets deposited, and this is the important feature of alkaptonuria, and we call it as ochronosis. So deposition of homozynic acid in the soft tissues and joints, so leads to condition called as ochronosis. So coming to another inborn error of amino acid metabolism, albinism. So albinos means white, colorless. So here there is hypopigmentation, so that's why we call it as albinism. And what is the incidence? Incidence is 1 in 20,000 births is in, that is the incidence of albinism. And the enzyme deficiency in case of albinism is thyrosinease. So in the metabolism of phenyl and tyrosine, we get an enzyme called as tyrosine. So tyrosinase is used in the synthesis of a pigment called as melanin. So melanin pigment deficiency leads to albinism because there is deficiency of tyrosinase. So without tyrosinase, tyrosine cannot be converted into melanin. So deficiency of melanin is in case of tyrosine deficiency, that we call it as albinism. So there are two types of albinism, that is oculocutaneous albinism and ocular albinism. When both eye and skin manifestations are there, we call it as oculocutaneous albinism, and only when it is eye manifestations, we call it as ocular albinism. So what happens in case of albinism? As I told you, so melanin cannot be synthesized and that leads to hypopigmentation. That is important feature of albinism. So apart from that, there is photophobia. So they cannot go into sunlight. So there is photophobia in case of these cases. And apart from that, nystagmus is another important feature. So nystagmus is the eye manifestation, and decreased visual acuity, another important feature of albinism. And there is skin is very sensitive to UV rays, UV light. So these are the important features of albinism. The most important feature is hypopigmentation is in case of albinism. So here you can see in the pictures, there is eye manifestation, we call it, whenever there is only eye manifestation, we call it as ocular albinism. When both skin and eye manifestations are seen, we call it as oculocutaneous albinism. And next inborn error of metabolism, maple syrup urine disease. This is because of, this is seen in case of branched chain amino acids. So the inborn error of metabolism concerned with the related to the branched chain amino acids is maple syrup urine disease. So when you see, so this is because of branched chain amino acids, valine, leucine, isoleucine. So these are the branched chain amino acids. Whenever there is defect in the enzyme which is concerned with the metabolism of these branched chain amino acids, it leads to a most important disorder called as maple syrup urine disease. So here the incidence is 1 per 1 lakh births is in case of maple syrup urine disease, and the enzyme deficiency in case of MSTU is maple syrup urine disease is alpha keto acid dehydrogenase. So that alpha keto acid dehydrogenase deficiency leads to maple syrup urine disease. The corresponding alpha keto acids are accumulated, and that is the main reason behind this MSUD. And when you see the clinical features in case of maple syrup urine disease, so this disease starts in the first week of life, and those who are suffering from this maple syrup urine disease, they'll die within 1 year of life. So the important features of maple syrup urine disease are convulsions, mental retardation, vomiting, and acidosis, because there is increased keto acids, corresponding keto acids of each branched chain amino acid. That's why it leads to acidosis. So these are the important features seen in case of maple syrup urine disease. So those are convulsions, mental retardation, vomiting, acidosis, and that person dies within 1 year of life. So laboratory findings, what diagnosis of maple syrup urine disease is. So these branched chain amino acids, branched chain amino keto acids, those which are in excess because of deficiency of alpha keto acid dehydrogenase, so they can be identified by a Roux-Tara's test. So apart from that, what is the treatment in case of MSUD? So we have to decrease the intake of branched chain amino acids in the diet. So that's how we can treat these MSUD cases, maple syrup urine disease cases. Thank you.